Good afternoon, everyone. Welcome to the STEM Lockdown Digital School. This is grade 11 Life Sciences. Okay, my name is Onkarabetse Setswe, and my Twitter handle, guys, it's at Onkarabetse_S. Right, so guys, today we're doing a continuation from yesterday, from where we left off yesterday. Remember, we're dealing with energy transformations, and the biological process that we're looking into right now is photosynthesis. Right. Yesterday, before our lesson ended, there was a learner who said that they don't understand. I would just like that learner to please use the question and answer section and just write quickly what you don't understand so that we are all on the same page as we move on. All right, so just please quickly write what you didn't understand from yesterday's lesson so that we are together and we can move together, all right? And if you got clarity on what you didn't understand, can you also please indicate that on the question and answer section? All right, so to everyone who is new, who's joining us today, welcome. And those who were joining us yesterday, welcome back. Right, let's continue with the lesson. All right, another thing is that yesterday we started, I think it was question 2.3, and I started that question with the assumption that we are all familiar with experiment questions, because that question was an experiment question, right? An experiment was carried out, even the one that we did before, okay? And I didn't go through what you need to know with regards to practical questions and what they comprise of. So today I'm just gonna have a young recap of what we need to bear in mind when we do such questions, because such questions are found in every exam paper, every test that you guys will write, you will find these questions. And of course, every practical that you guys will do, these are the type of questions that you will find. All right, so the first thing is the aim. The aim indicates the purpose of the investigation. So the aim is usually blatantly written out, or you will find it in the description, right? And a question might appear asking, what is the investigative question for this experiment? And this is the part where you turn the aim into a question, all right? So you just turn the aim into a question and we keep it moving. Okay, then we have what we call the hypothesis. So what do you suppose is going to happen? It's an assumption, all right? It can either be positive or negative. By positive or negative, what I mean is it can either be correct or incorrect, right? It's a statement. What do you assume is going to happen once you carry out this experiment? Or rather, how would you answer your investigative question, right? Like answering an investigative question, giving a statement on answer to your investigative question. Okay. Then we have method and materials. What are you going to use? How are you going to perform the experiment, right? So your method and materials, they are an important section or part of the experiment because you need those for the actual experiment to take place. And then we have our results. What do you see? What do you hear and smell? Or if you're working on a question paper or a test, is what you would say is what was heard, what was seen, and what was smelled, right? So there are different ways to describe your findings. Either by a description can be given. You can have a diagram. An example is the question of the graph, right? The rate of photosynthesis against light intensity that we did yesterday, right? Have photograph, a graph. We have various types of graphs. I think that we are all familiar with the different types of graphs that exist. For example, your line graph, your bar graph, your pie chart, or a table, right? Then we have a discussion. The discussion part is where we discuss the results obtained in terms of how it answers the aim. And then we have a conclusion. This is a sentence or two stating whether the hypothesis was supported or not. And then you answer the question in the aim, right? The question in the aim is your investigative question, right? Okay, I would like to just state this again. The student that asked a question yesterday indicating that they do not understand, can you please write on the question and answer section what you did not understand so that as we move on, we can all be on the same page. Can you please indicate it there quickly? And if you are fine, just also please indicate. All right, let's go back to our practical investigations and experiment questions. So another thing that we find when we're doing experiments or practical investigations is your variables, right? We should be able to identify variables. So we have what we call the independent variable, we have what we call the dependent variable, and we have what we call control variable or variables, right? So the independent variable, this is the variable that you have control over. Again, it is the one that you change. So time is usually an independent variable. Guys, note that I said usually, not always. Usually, right? And with the example of time, scientists usually use the dependent variable, okay? And graphically, it is represented on the x-axis, okay? Then the dependent variable, this is the variable which is being investigated. This is the one that you don't have control over, or the person who is performing the experiment doesn't have control over. And graphically, it is indicated on the y-axis. Then we have what we call the control variable. This factor doesn't change, okay? It doesn't change at all during the experiment. It's something that is kept constant. I hope we're following. Okay, so remember we have what we call the independent variable, the dependent variable, and your controls. So guys, yesterday I did ask that we do this for homework. All right, so I will be expecting answers from you guys. And please use the question and answer section. And so we're basically marking, right? Guys, please use the question and answer section to submit your answers so that we see that we're on the right track. But nonetheless, I'll just start with giving out the answers, or rather, no, let me start reading out the question and then we can do the answers. Okay. So question 2.2 reads, when light shines on pondweed, Elodia sp, bubbles of gas are released. The rate at which bubbles of gas are produced can be used to measure the rate of photosynthesis. An investigation was carried out to study the effect of different colors of light on the rate of photosynthesis in the pondweed. The pondweed was exposed to one color of light and left for 5 minutes before measurements were taken. And then the time taken for the release of 20 bubbles was recorded. The procedure was repeated using light of different color or equal intensity. The apparatus was set up as shown in the diagram below. The results are shown in the table below. Okay. So guys, as you read your description, keep in mind what we just covered. Aim, hypothesis, discussion, results, and as you go through the question, indicate, okay, this is the results section of this experiment. This is the materials, the apparatus right there, the materials that we used. This is the method, right? And then you can jump into your questions. Let me just get a pen. All right. Question 2.2.1 reads, which color light is the best for photosynthesis? So now we need to go back to our results table, Okay, we can see that for here we have the color of light, and here we have time taken to release 20 bubbles, and the unit used is seconds. We can see that for the color violet, 80 bubbles were released. For the color blue, we have 40. For the color green, we have 160. For the color yellow, we have 140. And for the color red, we have 70. So when you go back to the question, it says, which color light is best for photosynthesis? Can I get answers, guys? What answer did you give to this? Let me just check the question and answer section. Blue. Okay, Danielle, thank you. Time in seconds is independent. Right, we'll get to question 2.2 right now. So yes, guys, the answer is blue. And the reason that the answer is blue is because released 20 bubbles in 40 seconds. So the color blue released those bubbles in the least time, all right, compared to all the other colors. All right, question 2.2, state the independent variable. Danielle just gave an answer. Okay, wait, I can't find, all right, there we go. Danielle gave an answer saying that question 2.2, the independent variable is time in seconds. Okay. All right, so for question 2.2, the answer is the color of light. Color of light, all right? And the reason for this, guys, let's go back to the previous slide where I explained the independent and dependent variables. Remember, I said that time is usually, not always, usually the independent variable. But in this case of our experiment, what we are changing as learners, or rather what was changed in this experiment, was the color of light. Okay, the color of light was what was changed. So it's independent. Okay. And remember, guys, here we're dealing with time taken to release 20 bubbles. The time taken to release 20 bubbles. So guys, when we are shining the green light on the pondweed, we don't know. We can't assume that the green light is gonna release 20 bubbles in 50 seconds or 20 seconds. We don't know. That is what we are measuring. So in turn, as we get to the dependent variable, we know that it is the time taken. Okay, where's that pen? Right. So this is the dependent variable is the time taken. It's this part here, guys. It's going to take me forever to write because, yeah, my handwriting is very crappy. But yeah, guys, the dependent variable is the time taken to release 20 bubbles, right, per second. I hope we're together. Danielle, do you understand why time in seconds is not independent? Okay, I'll be waiting for your response as we move along. So question 2.2.3 reads, calculate the average time taken for the release of 20 bubbles. Average time taken for the release of 20 bubbles for all colors. Show all your working. Guys, this last statement here is important because that means you will get marks for showing your working and not just typing into your calculator and giving an answer. All right, so what we need here firstly is a formula to calculate average. Okay, I'm just gonna write the answer here on top. So the formula to calculate average, or rather how we calculate average, we could say average, I'm gonna use AV, okay, is equals to the sum, okay, the sum of all the numbers okay guys, this hashtag is numbers, indicates numbers, okay, divided by the number of groups, right? Divided by the number okay. So that is the formula for average, right? The sum of all the numbers divided by the number of groups. So now when we make it relevant to our study today, the number of groups that we have is 1, because we have violet, blue, green, yellow, and red, and this is 1, 2, 3, 4, 5. Okay, so it's going to be 5. That's the number of our groups. And then we can see that the sum, it will be 80. Plus 40 plus 160 plus 140. Oh, sorry guys. Plus 70. Ooh, I don't know how that's gonna fit. Okay, but there we go. Plus 70. Okay. So we're gonna add those numbers up and divide them by 5, and our answer is 98. Okay. So that equals to 98 seconds. Oh, wow. Guys, please remember not to write like me when you answering tests, exams, and assignments. Thank you. Anyways, yes. So we're at question 2.2.3. So guys, you will get a mark for this. You're working out, right? You will get a mark for the answer, and you will get a mark for your units. Hence, the mark allocation is 3. Okay. So question 2.2.4. Right, that's correct, Danielle. You got it correct. Okay, question 2.4 reads, express bubble production under violet, blue, and green light as a ratio. And the mark allocation thereof is 2 marks. So we'll be looking at violet. We know that violet is 80. Okay, it's a ratio, remember? Then we're gonna, oh my goodness, there you go. Guys, it's not an exclamation mark. I was trying to make a colon. And then we have, we required blue, which is 40. Okay. Took 40 seconds. Sorry about that. Then we have green light, which is 160. Okay, so guys, as we can see from the numbers that we have, we can, the common denominator for each would be 20. So we'll divide, no, actually the common denominator is 40, yeah, it's 40. So 40 can go into 80 twice. Okay, sorry about that. Let me just get my eraser. Okay. Forty can go into 80 twice. 40 goes into 40 once, and 40 goes into 160 4 times, right? So that is our answer, and it is 2 marks. If you left your answer like that at 80, 40, 160, you'd probably just get 1 mark, right? Okay, so question 2.2.5 reads, explain why the apparatus is left for 5 minutes under each color of light before taking measurements. So guys, the reason for this is to allow the plant to adjust, okay? And it's basically adjusting to the new rate of photosynthesis, because the rate of photosynthesis under violet light is not equal to the rate of photosynthesis that will happen under blue light. Okay, so that is just basically to help the plant adjust to the rate of photosynthesis. That is why the apparatus is left for 5 minutes under each color of light before taking measurements. Okay, question 2.2.6. Without modifying the apparatus, how could the reliability of the results be increased? Okay, guys, this is an easy one. Okay, repeat the experiment. As when you repeat the experiment, you will measure your results that you got from the first experiment and from the second experiment. And again, you can also do it for the third time. Repeat it for the third time and get the average of the results that you got from all experiments. And that will increase the reliability of the results. Okay, and this is without modifying the apparatus. Question 2.2.7 reads, using the results, explain how when white light shines on a plant, the leaves appear to be green. Wow, guys, question 2.2.7 actually requires previous knowledge. Okay, knowledge that you got from grade 8 and 9 natural sciences, where you were dealing with light, where you were dealing with the reflection of light. Okay. And yeah, so basically to answer this question, you need to have a strong background of your biological sciences. But it's not difficult, guys. It's really not difficult. And I just want to use this diagram to explain it. So guys, let me just get a laser. There we go. So when white light comes in, right, it hits the leaf there, and then green light is reflected. Okay, you guys remember the properties of light. There's refraction, there's reflection. So in this case, when white light hits the leaf, okay, the green light is reflected, and hence we see the green color of the leaf. So basically, to answer this question, it's just to say that more green light will be reflected by the leaves, or green light, or you can say green light is poorly absorbed by the leaves compared to all the other colors, because white light, guys, is a spectrum of colors, right? And then the leaf absorbs all the other colors but reflects the green. All right. Okay, let's go back to our question. The last question reads, draw a bar graph of the results shown in the table. Okay, guys, I have the answer of the bar graph on the other slide, so you guys will just compare, and I will go through it with you as to where the 8 marks goes. All right, so first thing that we need to understand here is that you are asked to draw a bar graph, not just any other random graph, but a bar graph. So here we have our bar graph. Bar graphs require a title. They require a title where you indicate both your dependent and independent variables. All right. So you will get a mark for that. And then we need the correct type of graph. You see, as we can see here, it's bars. So you have 1 mark. Then we need correct labels for the x-axis. There's your x-axis, your independent variables. That's another mark. And you need the correct label for the y-axis. Y-axis, there is your label and your units. Okay, units are also necessary, guys. That's your other mark. And then you need an appropriate width and intervals of bars. So basically the distance here, or rather let me use it here, the distance here and the distance there need to be equal. Okay, and you get 1 mark for that, and you also get another mark for an appropriate scale for your y-axis. Okay, and the drawing of bars. If your bars here for Violet are 80, you know, they correspond with the results, you get 2 marks. Okay, so that's where all your 8 marks go for that question of drawing the bar graph. Okay, guys, let me just quickly check questions if we have any questions. Okay, so we don't have any questions so far. Guys, are we all on the same page? Do we all understand what we're doing? If there's anything from the previous lesson that you didn't understand, can you please just type it down so that we go through it? But if we're good, then we can move on. Right, cool. Okay, so just as I had mentioned that in all biological processes, basically, we have various experiments to test whether the biological process took place. So right now, we're just going to look at various experiments related to photosynthesis that can be carried out. So basically, what we'll be looking at right now is some of the type of questions that you can get in tests or exams relating to photosynthesis, or rather testing if photosynthesis took place. Okay. Right. Let's revise quickly. Let's go back to, I think it's grade 10. Yes, testing for starch, grade 10. Remember, guys, in grade 10, we had practicals where we would test chemicals. I think others use potatoes to test for starch, right? So the chemical we use to test for starch, let me just get this quickly. Change the color. This color is actually very horrible. Iodine solution. Wow. Blunt moment. Sorry. Oh my gosh, worse. Wow. Okay, there we go. Iodine solution. Right. So the chemical that is used to test for starch is iodine solution, and we know that if the test is positive, or if the liquid that you're testing or the solid that you are testing has starch, the color will change to blue black. Right, so that's very important. That is something that you need to note as you continue with photosynthesis, because the starch test is very important in testing whether photosynthesis has taken place in a particular plant or leaf. Okay, so now, because we are talking about testing for starch in relation to photosynthesis, let's go back to our first lesson where we said that photosynthesis, we need carbon dioxide, we need water, we need light, chlorophyll, and enzymes to produce glucose and oxygen. Glucose and oxygen are the products of photosynthesis, and glucose is usually stored as starch in the leaf. Right, so that is how we can measure if photosynthesis took place, right? So how do we test for starch in a leaf? This is how we do it. So firstly, we'd heat the plant leaf in boiling water for about 30 seconds. The reason for this step is that it stops chemical reaction to take place in the leaf. All right, and then we place the leaf in ethanol or methylated spirits, and alcohol basically, to break down the chlorophyll. And then we wash the leaf with water and spread it onto a white tile. White tile does not necessarily mean the tiles there in your house. Even glass slips, can just put it there so that it dries up a bit. And we add the iodine solution from dropping pipette. And basically, that is how we test for starch in a leaf. So guys, if we come across in the next coming slides saying that perform a starch test, this is basically what it's referring to. Okay, this whole step. This is how you perform a starch test. Okay. Check for any questions. No questions. So I guess we're all good. So right, destarching plants. How and why? Why should it be done? Destarching a plant, it involves preventing photosynthesis. So it's usually done by keeping the plant in the dark for at least 48 hours until the starch that is stored is used up. Okay, so this is also a critical step in all investigations, in most investigations pertaining to photosynthesis. And then it is necessary to destarch a plant before performing photosynthesis experiment because the amount of starch formed during the experiment should be validated. So guys, please just note this last point here. It is very important. And guys, in most of our experiments, you will see that they say that they put the plant in the dark for at least 48 hours. Again, remember the reason, it is to prevent photosynthesis. It is to prevent photosynthesis. All right. So here is one of the first tests that we have. The first one is to determine whether light is necessary for photosynthesis. This is a type of test that can be carried out, or experiment that can be carried out to determine whether light, the key word here is light, is necessary for photosynthesis. Remember that we said that light is necessary. So here we're testing whether or not the statement is true or false. So what we will do is we will put the plant in the dark for 48 hours. Why is that? This was mentioned on the previous text, on the previous slide rather. This is to prevent photosynthesis from happening, okay? And then what we will do is parts of the plant will be returned to the sun. What do I mean by parts of the plant? Just look at the picture here. So on the leaf, you will cover the leaf with foil, okay? You can either make this opening or not, it's fine. But you would just need to cover part of the leaf so that it is not exposed to the sun like this part, right? Because what we want to see is whether whether light is necessary for photosynthesis to take place, okay? So this is what the second bullet is explaining, that some parts of the leaf or the plant should be hidden from the sun and others should be exposed. And then what we do is we perform a starch test. Starch test, guys, remember from the previous slide that a starch test includes putting the leaf in boiling water for about 30 seconds, putting it in alcohol, and then pouring iodine on the leaf. So we'll pour iodine on this part of the leaf and pour iodine on that part of the leaf. And at the end of our experiment, we will see that the part which was exposed to light will have starch. So the color will turn to blue-black, but this hidden part here will not, the color of iodine will not change, right? So another test that we can have is to determine whether chlorophyll is necessary for photosynthesis. Okay, so we use variegated leaves, okay, and we put them in the dark for at least 48 hours. Remember that I said that putting the plants in the dark, this is to prevent photosynthesis from happening. So right, then the second bullet says then the leaves are put back into the sun for several hours, okay? And then we perform a starch test, okay? So this is what's happening. Remember, photosynthesis takes place in green plants. So if you have a plant like this where it has certain sections which are not green and certain sections which are green, it indicates to us immediately that by the green, where the plant is green, that is where photosynthesis will take place. Okay. So after we perform the starch test, remember starch test, put the leaf in boiling water, alcohol, and so forth. This will be our result. Okay. So our result, as guys we would have assumed, is that the green parts of the leaf have that color, the blue-black color, that indicates that starch is present and that indeed photosynthesis has taken place. But the color, the part of the leaf which was not green hasn't changed color to blue-black, as you guys can see in this picture below. So we can see that after performing our starch test, right? Another experiment or practical investigation that can take place is determining whether carbon dioxide is necessary for photosynthesis. So in this type of practical investigation or experiment, we need to be using sodium hydroxide or any other form or any other solution that has sodium hydroxide solution, guys. This solution part is very important. Don't just use plain sodium hydroxide, you will die. Please, sodium hydroxide solution. Okay. And what is the sodium hydroxide solution's purpose? This is to absorb carbon dioxide from the surrounding air. And another solution that we'll use is water or any other solution that is written out or that the scientists have used, okay, or that which you as learners at school have. Okay, so what will happen is we'll be using two plants. We cover both plants with a plastic bag and secure them with elastics. Okay, so guys, this experiment, it can take place in different forms. Okay, it does not necessarily mean that in every experiment to test for carbon dioxide, we'll be using plastic bags. This is just a blueprint, if I can call it that. Right, so we cover both plants with plastic bags and secure with elastics. I will show you guys in the upcoming slides a picture of that. And then plants are left in the sun for several hours, and then we perform a starch test. Okay, so this is plant A. Plant A has our sodium hydrogen carbonate solution there. So you see, guys, in plant A, they use sodium hydrogen carbonate solution instead of water. So you guys can replace the sodium hydrogen carbonate solution with water, and they placed it there, and that's the plant with the plastic bag, and it's covered with elastic. And in plant B, we have sodium hydroxide solution. This is the solution which the sodium hydroxide solution, it is the solution that absorbs carbon dioxide from surrounding air. Okay, so our results will be like this, that the plant that had sodium hydroxide with it in the plastic bag will produce less starch because the sodium hydroxide would have absorbed the carbon dioxide that is in the air, and then the plant that had sodium hydrogen carbonate or water with it in the plastic bag will produce more starch, which will be plant A. All right, so that is how we will be testing whether carbon dioxide is necessary for photosynthesis. How will we know? We perform a starch test on the leaves of the various plants. Okay. Move along. And another example that we have is determining whether oxygen is produced during photosynthesis. This experiment, guys, it is similar to the question that we just went through. All right, so the bubbles that form there, those are indication of oxygen that is released. All right, so yeah, to determine whether oxygen is produced during photosynthesis, we can just refer back to the question that we did, and we'll get clear answers on that. I don't know if you guys are, so okay, another thing that I need to mention is that in this particular experiment, there is no starch test which is being performed because, of course, what we need here is bubbles. We measure this by the bubbles that are released. Okay, let me check if we have any questions. Okay. Kabishi, why is the plant left on the sun for hours? Right, Kabishi, I hope I'm pronouncing your name correctly. Sorry if that's the incorrect pronunciation. I think you're referring to this one, right? To this particular test that plants are left in the sun for several hours. Okay, so the plant is left in the sun for several hours so that photosynthesis can take place for this particular experiment. All right, remember that in order for photosynthesis to take place, we need light, we need sunlight, right? So that is why the plant is left in the sun for several hours, so that the plants can absorb the sunlight and photosynthesis can take place. Yeah, that is why the plants are left in the sun for several hours. I hope that answers your question. Okay, so we don't have any more questions. All right, guys, let's move right along. We dealt with this one. Okay, so guys, I have a question for you guys. This is just additional practice questions that will follow. I need you guys to take a screenshot of this. If you're using a desktop, just manage or use a snipping tool. Using your phone, just take a screenshot of this. And again, guys, this video will be on YouTube, so you guys can go back and watch the video on YouTube if you missed some of the questions or some of the explanations. So guys, okay, this is question 3, right? It's actually similar to the question that we did yesterday, if I'm not mistaken. Yesterday, yes. It's similar to the question that we did yesterday, and I need you guys to do it on your own, all right, and send me the answers. I will give you the email where you guys need to send the answers. Okay, so this is basically, it says that the diagram shows a plant propagator in which scientists can control temperature and light intensity and carbon dioxide concentration, and there you have your diagram. And scientists set different temperatures. Here you have your method and your results. Okay, and the questions are as follows. What is the influence of light intensity on the mass of lettuce plants? We dealt with influence of light intensity and the rate of photosynthesis. Name the 2 limiting factors that influence photosynthesis as light intensity increases. How did the scientists be able to increase the rate of photosynthesis to maximum level? The last question, what would happen to the rate of photosynthesis if the temperature is raised beyond 35 degrees Celsius? Give a reason for your answer. So guys, all these questions, they are related to what we have covered throughout our lessons of photosynthesis. Okay, let me just get this quickly. All right, cool. And then here is another question, which is also very important. Can you guys please also take a screenshot of this, or if you are able, just watch the YouTube video again and you can pause it there. But just preferably take a screenshot. And yeah, so in this diagram, the diagram below shows the apparatus that was used to demonstrate that CO2 is released during cellular respiration. The apparatus was set up as shown in the diagram and placed in a dark cupboard for a few hours. A suction pump was connected at the outlet marked X. So guys, don't mind that cellular respiration, okay? Take it as photosynthesis, because that is what we are dealing with now, photosynthesis, right? So yeah, photosynthesis, because in a way they are similar, but just read the cellular respiration as photosynthesis, right? And yeah, answer the questions which are below. What is the function of the soda lime? Okay, let me just quickly explain soda lime. Remember on the other slide where we spoke about sodium hydroxide? So soda lime has sodium hydroxide in it, right? So soda lime is a substitute for sodium hydroxide solution. Okay, why was the apparatus placed in the dark? Why was the glass tube marked Y placed over the surface of the solution? And after a number of hours, what observation would you expect to make in bottle B and D? Explain your observations. Right, guys, so please take screenshots of these questions. If I need to go back to any questions, just please say. Okay. And guys, take away message for this topic. Know your terminology. Remember, guys, terminology is very important. So know all definitions that were covered here. And also, guys, make sure that you meet all the learning outcomes. I I went through them with you guys on the first lesson. Make sure that you understand all concepts that were covered. For example, the effects, the factors that influence the rate of photosynthesis, guys, being able to draw the chloroplast and being able to label it, knowing the functions of the certain parts of certain components of the chloroplast. Please, guys, it's very important. And also the practical investigations that we went through today, because as you guys have seen, most of the questions, they are practical investigations, right? And they ask you concept related questions in the practicals. Okay. And another thing, guys, these two equations, very important, the word equation and the chemical equation. Remember, we need 6 molecules of water, 6 molecules of carbon dioxide. Light, chlorophyll, and enzymes are necessary in order to get your glucose and your oxygen. So guys, you might be asked in tests and exams, write down the chemical equation of photosynthesis. This is the equation that you will write. Or you'll be asked to write down the word equation of photosynthesis. This is what you will write down. Remember that this side of the arrow indicates your reactants, and this side of the arrow indicates your products. Okay. So guys, some other helpful links. You guys can follow Teaching Biology Project on Facebook for more insightful info. And yeah, this is the email address that I need you guys to send your answers to the previous questions that I asked you guys to do. So you guys can send your answers through to africa10geeks.onmicrosoft.com. Okay, and then I'll give you guys a response from there. Guys, remember to follow Africa 10 Geeks on all the social media platforms, at africa10geeks.co.za on Facebook and on YouTube. Thank you guys for joining in on this lesson. And I hope you guys learned a lot. Hope to see you. Oh, sorry, Kensani, may you please go back to the first question? All right. I'll do that quickly. This one, there you go. I hope that covers you, Kensani. I hope you covered Kensani. All right, guys, thank you very much for joining me today. Remember, tell your friends to tell their friends to tell their friends to join us tomorrow again for grade 11 life sciences. Tomorrow we'll be dealing with animal nutrition. All right, guys, have a lovely day further. Bye.